Derya, thank you so much for being here.
Thank you. I'm very excited.
Likewise, very excited to have you because you're obviously a very unique kind of builder, quite unlike the builders we usually talk to. You have a medical background, but you're also working in bioscience, bioengineering, and you're pushing in AI in a way that like most builders don't. Coming at it with this real depth that you have in so many topics, like from biology to cancer to immunology. So very excited to dive in.
Thank you. Thank you very much.
Maybe the first question, like, if we rewind, like, when did you first realize that biology and science were going to need AI?
Yeah, I think that was right after I graduated from medical school, when I realized the complexity of the biological system. In fact, after I graduated, I went into biomedical research because I really wanted to understand the biology. And I saw that, at the time, we couldn't treat a lot of diseases and because of the complexity. And then more I went into the research aspect of it, the more I realized, oh my God, this is incredible. How are we going to solve this? There are trillions of different parts in biological systems and billions of reactions that are happening. And it just seemed so overwhelming. And that's when I got interested in AI, artificial intelligence, early 90s. Then I realized maybe someday, we could actually build models using AI, and got very interested in trying to code with AI during the 90s. And of course, fast forward with deep learning revolution, then I was so excited because for the first time I could see that deep learning is able to process this massive amount of information in a sort of a parallel fashion. And of course, AlphaFold and then ChatGPT. Yeah, but the first moment was when I finished my medical school.
Amazing. And since that moment of launching ChatGPT, it feels like you've very much been in our community, right? You've been testing models. I remember you doing some work with O1 Preview.
Yes.
When we had the very first reasoning models, what was the first reaction when you started to have your hands on that?
I still remember this was September 2024. Actually, OpenAI approached me, I guess because I was very active on X and or Twitter in the old days, talking about AI, how this was going to change humanity. At the time there were a lot of skeptical people, and I think there still are. But I truly believed in it, and I went all in AI. And I guess OpenAI was interested for me to try. I guess the first reasoning model, and I still remember it when I gave it a very complex question on immunology in my topic. But the question, actually, I even remember the prompt. I'm very interested in games, video games. So I like to sort of cross-fertilize games to science. And one of the games that I would play is, this sort of like a survival game when you're in an island and you're fighting. I won't give the names of the companies, but you understand the type of games. And so it's kind of a battle royale games. And in a way, the immune system is kind of like a battle royale against tumors. And I said, think of how would, if you just imagine battle royale games and the immune system, how would you develop a scenario where the immune cells would fight against cancer? It's a completely different field. And in fact, we actually did experiments related to that later on. And O1 Preview just gave me this response that almost made me emotional. I was like, oh my God. Before that, GPT-5 4.0, it wouldn't really give you that kind of a deep, insightful response. I still remember that day. That was very special.
Was that the moment you'd say where you thought, okay, like AI is now inevitable for science?
Absolutely. I mean, even before that, especially after GPT-4 came out, it was extremely useful. I would tell my colleagues, because there's so much information, especially in biology, you can't really keep up with it. And for searching literature and really synthesizing the knowledge. Of course, doing mundane things like writing your letters. I write a lot of letters of recommendation, and things that we used to take an hour would take 5 minutes. but it wasn't at the point where you could fully trust it or ask questions like, so what would be the outcome of an experiment, for example. And I think O1 preview was that moment where I realized when it starts to reason, then you get things that's going to be extremely relevant for science. Of course, after that, we had the Pro version and O3, which got better, and now we have unbelievable models along. And that, I can mention about my current experience, and which is getting better and better.
Yeah, I mean, like maybe speaking of the current moment, like fast forward to today, I read a couple months ago your tweet about your new routine where like you're gonna start your days now with first a morning coffee, but second, kind of Codex has to be doing something for you.
Yes, so I can call myself a Codex addict. I'm definitely a certified one. Yeah, so the first thing in the morning when I wake up, because what Codex enabled is, there's all these ideas that, whether you want to make a simulation, whether you want to make a certain app, or you want to make a game, or whatever it is. In the past, you had to know how to code. Even if you knew how to code, it would take you weeks or months to do it. But now, I get a lot of ideas, and the moment I wake up, get my coffee, say, okay, I got to try this idea. How is it going to work out? And sometimes, actually, Codex is working overnight, so I want to see what the results are. And I haven't slept that much in the last couple of months because of that.
Fascinating.
So yeah, I love Codex.
And I'm curious, like for someone like you that has so much like real depth in like immunology, oncology, cancer, T cells, like how do you approach like using Codex to kind of bring these worlds together in a way that's also useful to your day to day?
Yeah, I can even show specific examples. I've been trying to build, some apps that are not very complex, but extremely useful for day-to-day basis. I have a bunch of examples of that. But then also we are very much dependent on software for analysis. As I said, biology is very complex, whether it's genetics. In immunology, we, for example, do a lot of analysis called flow cytometry. Basically, this is like our window to the world of cells, and mainly immune cells, but you can analyze any cells. So we have these machines, there's a big laser. We label the cells with particular markers, fluorescent markers, because there's like hundreds of different types of cells. So how do you know what type of cells there are in your blood, in your tissues? So we label them, we pass them through this laser, and the laser, analyzes thousands of cells and then creates some sort of a data that will tell me this is an immune cell that can fight against cancer, this one can cause autoimmunity, or whatever the function is. But we have to take that data and put it into specialized software, take all that thousands or sometimes hundreds of thousands of data points, individual cells, and the software will make that into a graphs, and then so you can say, okay, give me the percentage of these cells there, and how do they relink to that, and so on. So this is very sophisticated software, which we've been using for the last couple of decades. And one day I said, why not make one for myself? It was a crazy idea because it's very complex. In fact, I failed a lot. I tried to make some things worked, but since 5.5, now I have a completely working version of this.
That's amazing. I would love to see it if you have like things on your laptop to show me.
I can show you some of these examples. So start with this. I have different versions of this, but I'll show you one of these applications. So basically, I already uploaded a file, and you can see that these are actually, each of these dots is a single cell. Mm-hmm. And these are the fluorescent molecule colors, if you like. So whatever the antibody has that fluorescent molecule, it will label a particular cell type. And actually, I can choose here, okay, well, change me, because you can see all of these are different antibodies that are labeled differently. And it's very complex because there's 20 different molecules, and each of them binding to a receptor, and their combination will give you a particular subset of cell that will be defined by those markers alone. For example, my favorite cells are these these T cells that have what we call the CD4 molecule, and then some of them have what's called the CD8 molecule. The ones that have CD8 are killer cells. They will go and kill the lymphocytes. And so I can put gates here, and I can say, okay, well, what's the percentage of cells that are CD8 positive or CD4 positive? So it will create all kinds of statistic analysis. But this is actually really complex software, because I can change the contour plots of the graphics. It's a different kind of representation. Remember, there's like 100,000 events here.
Wow.
And it's doing it so fast, which is incredible, because I didn't think that it would be optimized this way.
And you built all of this with Codex?
100% with Codex, yeah.
Incredible.
I mean, it took me a while, because some things wouldn't work, but especially with 5.5, I would say, okay, well, I can't see the graphs very well, and fix that and write me a prompt for that, and it would just go and work. And So I made this little app, which basically you can actually choose the cell type you want. Okay, so I say I want a naive T cell, and it will show me all the markers that I can potentially choose. And in fact, it will even show me, for example, the ones that are more relevant for that cell type versus the other one. This becomes extremely useful for developing these what we call panels. I'm looking for the central memory naive cell and a T cell and Th17 cells. Okay, if I choose these markers, then I can go back and do my flow analysis.
That's amazing. I mean, you're obviously an engineer by training, but it would have taken you, I'm sure, weeks of work, if not months of work, to even build these apps, right?
So I'm not a software engineer. I'm a biomedical engineer. So
sure.
I can't really write. I could probably write a snake game if I worked for a month. To make these apps would be kind of like a dream. So, I mean, I make these little kind of apps that help on a daily basis.
That's pretty incredible. And on T cells, tell me more. Have you try to play with more models to kind of help in your day-to-day work using Codex or even things like ImageN?
Yeah, so one of the things that I'm very passionate about, and I think when I say AI is going to make tremendous impact on especially biology, is to be able to simulate biological systems because they're so complex. For example, if you're building an airplane, right, you don't just say, okay, if I put these parts together, I hope that I'm going to have an airplane. You do a simulation, right? So how is it going to be aerodynamic and all that stuff for any kind of complex things. But for biology, we don't have that because there are so many parts. And my goal is that at some point we'll be able to build what's called virtual cells. We will be able to mimic a complete immune cell by AI and eventually tissues, and eventually what I call the digital twin, like a complete human being. We're going to need a lot more compute, so you guys better invest developing that.
Point taken.
But
okay.
Just to start, I began with an app like this. This is basically one receptor. It's called the T cell receptor that's on the top of these immune cells, the T cells that I just showed you, and it's the main receptor. But one receptor is extremely complex. Depending on the affinity of the molecules it sees, depending on the other signals it sees, that's the decider. That will decide sometimes whether you're going to live or die, because the strength of the signal could mean that you get autoimmune disease. It could mean that you can clear the tumors. It could mean that you kill the virally infected cells, or it could mean it will cause too much damage and kill you. So the decision point is there, and there's very complex events that happen underneath that receptor. There's huge signaling pathways. And so I built this just to be able to simulate. For example, if I have only the T cell receptor, and then if the ligand is a certain quality and the dose is this much, I can actually control all of that here. And then I can say, okay, run me a simulation. Based on that, it will run me a simulation. It will tell me what molecules are going to get activated, which ones will not get activated, and it will even show me the phosphorylation patterns of what's called the transcription factors. And I can say, okay, well, what if I just put a pressure, there's an inhibitory molecule, and then I choose a different signal, what would happen? And then it will show me, okay, this pathway is stopped now, so you're going to get a different type of events. And so basically, you can just expand this. You can say, okay, what if I just put a small molecule that will inhibit this molecule? What will be the output?
Right.
And then what if I just add more receptors to here? And how are they going to interact with each other?
I love that because it's not even just like visualizing data sets or like searching in data sets. This is like a complete like application that you build to like identify the inputs and outputs in the browser of every like particular like cell and scenario. Incredible. I mean, we see so many builders who are obviously like using tools like Codex and GPT 5.5 to build like websites, web applications, mobile apps, but this is like so intricate and so advanced for the biology field.
Yeah, that's why I wake up early after coffee. I say, okay, so I'm going to add this to this simulator, see what happens, and then the other thing, which was kind of a fun thing to do. Because after Image 2.0 came out, I just fell in love with it. You know, I can make extremely complex images. For example, this image, I think I shared it on X as well. This was about an immune atlas that was published in this journal called Nature, it's the top journal in the world. And so I basically asked Image 2, said, you know, create me a cover page of Nature just for fun based on this atlas. And it made this unbelievable image where you can see all these immune cells are the ones that I've actually worked for the past 30 years. So I was like, oh my God, you know, this is so detailed and so beautiful. And then I said, what if I just take that image and put it into Codex and tell it to sort of make it live in an interactive website, and that's what I did. And then Codex just came up with these beautiful animations and how these cells are interacting with each other. I can click on them. It shows me what, you know, this is a memory cell. This is an effector cell. It gives me all kinds of information about them. You know, if I block it, for example, this molecule PD-1 is very important in cancer therapy. If you block this molecule, you can cure a lot of cancers, called checkpoint inhibitor. And, you know, this was totally a fun thing to do, but then I realized, wow, I can even change, for example, based on your age, how your immune cells are going to behave. This is like a mini simulator here. It all started with just one image.
Just this one image, that GPT Image 2 generated.
Exactly. And I didn't say, okay, do this or do that. I said, I want you to understand this image and create an interactive simulator website. That's all I did. And Codex just built this for me.
Amazing.
Yeah.
It's also such a great tool for education as well, right? Because like if you want to learn into like dig into any topic, learn about that topic, you can now generate image, but also just generate like simulators for that topic. It's pretty incredible. Yeah. It sounds like you're building so many apps. Like, thank you for sharing. This is like a window into your work and how you think and how you use Codex in a way that like I've not used Codex to build these apps so far. So this is fascinating for me.
Let me just show one more app, which I think is very useful. As a biomedical engineer, what we want to do is we want to manipulate the cells. In a way, the cells are kind of like programmed code, software. And I'm hoping that one day we'll have a Codex for biology where we can totally program the cells. In fact, we started doing that with these technologies to genetically edit cells. I actually developed some of that 25 years ago, 30 years ago. And now we have this thing called CRISPR. I'm sure a lot of people have heard of it. Basically, CRISPR is you can target any gene, you can change fixed mutations, you can delete the gene, you can overexpress it. It's like a genome engineering. But the problem is that, again, this is very complex. So if you have a gene that's, let's say, 2,000 words, nucleotides, where are you going to target? Right. So you have to have some sort of a computation to say that that's going to be specific, it's going to do this effect or not. And there are some tools like that. I said, well, I want my own, you know, so that I can. So I built this application where you can actually choose any gene you want. So let's say this is a CD4 gene, which is, I mentioned about that on the surface of T cells. It immediately pulls the sequence from the database.
So that's the DNA of the CD4?
Yeah, exactly. And then it gives me all the potential targets. So these are 22 nucleotide regions, but it's a very big gene. And then it ranks them. It says, okay, well, you should probably choose this one because this is good. And I can just say, okay, add that there, and then I can copy it and then give it to an online company, and they will synthesize that and send it to me, and I will engineer and do the experiment. I can visualize that. It shows me all the regions where these are being targeted. So there are some features here that are not present in other applications. That's the beauty. For example, I said, okay, can you create a library for me? So what if I have, multiple genes and I want many different CRISPR targets? So I built this version where I can just write the gene's name here, and all I do is just click on Design Library. It will make different CRISPR molecules for me. And
that's amazing. And this is a native app that we're looking at on your Mac, right?
This is a native Mac OS app that's built with Swift, and I'm going to make an iPad version too, so that I can carry it inside the lab.
Well, thank you so much for sharing that. This is incredible to see the behind the scenes of your work.
Yeah, now you understand why after coffee I start.
I totally understand the routine now. It makes so much more sense to me. One thing that you mentioned earlier that I'd love to double click on is this idea of like a digital twin, right? And as you have like your vantage point in like immunology, cancer, biology, when do you think this idea of a digital twin becomes viable?
First of all, why digital twin and why we need AI? The reason we need a digital twin, and when I say digital twin, I mean a person's complete biological system. So our biology is not just what you measure outside. As I showed you, there's the immune system, there's our metabolites, there's our trillions of bacteria in our gut, and hormones, and so on and so forth. And it's a very, very complex system. And of course, there's your genetics. And your genetics and your environment decide pretty much everything, whether you're going to get sick or when are you going to get sick, whether you're going to respond to a given treatment, can we predict before the disease happens? And it has to be very personalized. In fact, you have to treat the patient, not the disease. But because the biology is so complex, we have been giving the same drug with the same disease to millions of people. For example, statins are used by millions of people, but it only affects a small portion of them. So can we come to a point where AI can completely simulate the biology and your biology with your genome, with your metabolites, with your proteins, your immune system? And if we can get that point, then we can start to ask this question: what if I make this change in my health? Or what if I give them this drug? Or maybe I will basically order a treatment just for you based on your biology, what the AI is going to tell us. And so we're getting into a point of complete personalization where the drugs will have nearly 100% effect with 0% side effect. And that means that the clinical trials that we do are going to be accelerated from 5-10 years to maybe 5-10 days. Literally, AI is going to do the clinical trial for you. And that's the incredible acceleration. Because when I say in the next decade or so, we're going to treat all of the diseases, eventually within 15 years, we're going to reverse aging and people will be able to live hundreds of years. People say, well, that sounds crazy. That's science fiction. It took us 50 years for cancer just to be able to treat a little bit. What they don't calculate is that AI is advancing exponentially. And this is also assuming that the compute is going to greatly increase, because even the current available compute, if you put all of the compute in the world, is not enough to simulate biological systems. There are trillions of components. So if we get to that point, which I believe we'll be able to do in the next 5-10 years, and we'll have the superintelligence then basically we simulate this digital twin with AI, and then we do an experiment, not on humans, but with AI for your biology, and that will change medicine. That will change everything.
So if we take the example, for instance, of like a cancer patient, say, and if we had their digital twin, what could a doctor do differently versus what they do today? Would they just basically try different hypotheses and experiments on that digital twin and see how that reacts, and that's how we treat the stable group and the treatment group almost?
Exactly. Actually, cancer and oncology is as close as we are today for personalization, because even the same cancer type have many different mutations. And so you probably, if you are a lung cancer patient, the first thing your doctor oncologist would do is to sequence the genes that are mutated, because depending on which gene is mutated, you have a different drug for that. For example, 1% of lung cancers can use a particular drug. And that drug is very effective in that 1%, but it's not effective at the 99%. So these are smart drugs that companies are developing. So we can come to a point where we can literally create a drug for all the mutations that you have. In fact, there was a case from Australia. This computer scientist using ChatGPT and Grok, created an RNA vaccine for his dog.
Right? Yes, yes.
So that's as personalized as you can get, because it was that RNA vaccine was created just for that dog's cancer's mutations. And there's a lot of trials going on with that. So I think it will change completely because, as I said, based on not only your genes and mutations, but your immune system. For example, immune system is extremely effective in killing cancer cells. And this has been a revolution, what we call immunotherapy, but it doesn't work in everybody. Why is that? Why is some people's immune cells able to recognize the cancer and kill, but others don't? Some of them become exhausted and whatnot. So if we could figure that out, and of course there are also side effects, because the immune system is very dangerous. If you activate it too much, it can actually cause a lot of harm.
I'm curious, like you seem so AI peeled and so Codex peeled, while also being like an MD. Like, what are the people around you thinking about AI? Like, do you try to get them also like to adopt these tools as fast as you are?
Yeah, I try very hard, and I think they think that I'm totally crazy. Although I think nowadays they're starting to see the potential. And I've been saying this since ChatGPT 3.5 came out. I think people are very hesitant. And I can understand that, because this is something so new that human mind is not able to comprehend this exponential advance. For example, people use GPT-4o a year and a half ago, which is like ages in AI world, and say, well, it was hallucinating too much. It didn't really answer to me very well. In fact, even the difference between GPT 5.4 and 5.5 is day and night. If you constantly experiment and then assume that it's going to get better and better, I trust AI models even for things that I've been working on for 30 years. Because recently, GPT 5.5 Pro model gave me this report that I almost cried. I said, how is this possible?
I remember seeing your post online about that experience.
Exactly, because that latest model crossed the threshold. Of course, GPT-5 and Pro and 5.4, they were fantastic, understanding the knowledge and the patterns and putting them together. But what 5.5 did was almost as if it had the same experience that I had working in the lab for 30 years, because there are certain things are intuition. You just know. It's not out there in the literature. You feel that, for example, I would bet with my students, I say, do this experiment, I'll bet you it's going to work this way. And they would bet sometimes, and they would lose 100% of the time, because, I just know that it's going to work.
You've had so many reps that you have your intuition.
Yeah, somehow that you have that intuition. And that's what I felt with 5.5. That it predicted the outcome of an experiment, very complex experiment we had done at 100% level, which was incredible.
And I'm curious, like if we continue on this rate of progress that you're also experiencing on your side, what do you imagine like your day-to-day looking like in a few years from now? Like what will be like unmistakably different for people like yourself and researchers around you in this world of biology?
Well, again, some of the things I say may sound very radical, but fortunately for me, but maybe unfortunately those who don't believe in this, is that there's going to be a complete fundamental shift In fact, I call this science 2.0 or 3.0. The way we do science is going to completely change. The days where we would, for example, come up with an idea, plan for experiments for weeks, and then analyze them for months, those days are over. We have to, and students or other scientists have to realize that we are in a very accelerated time scale, that the way we do science is going to be a bunch of AI agents that will come up with the hypothesis, and they're already able to come up with the hypothesis. Because again, the number of things you can come up with are almost endless. How do you know that hypothesis is better than that one? Then, you'll get the best ideas. Then it will simulate the experiments for you. Because again, I can do 1,000 experiments. Well, which one's going to work? I don't know that. If it could drive me to a point that says, okay, this 10 experiment likely to work more because of this and this, then I can just focus on that. The chances of that experiment working is going to increase tremendously. And the data I get, I have almost instant response. I'll give it to other AI agents, and they will immediately analyze and feed it back to the master, and that master is going to make a new hypothesis and plan for a new experiment. So I guess my role is going to be just telling the agents, go figure this out. This is the direction you should go. I want to cure lung cancer and work on that.
Go explore.
Exactly. And then, somebody still has to do the experiment, but then I think the labs are going to get automated. This is already happening. And so there's going to be a bunch of robots doing lots of experiments. And people say, well, but will I still have a job or whatnot? There's something called Jevons paradox. If we are able to do so much, then we will be able to do so much more, especially in biology. As I said, we can get to a point of bioengineering, but we only know like 10% of biology. Imagine how much more, how fast we can learn. And then once we have that power, like I'm building apps now, then I'll be able to build new cell types, new tissues. So there will be maybe thousands of bioengineers, in front of their computers simulating and building. But this is going to fundamentally change not just biology. I think every field, physics and material sciences and chemistry. Already for drug discovery, you can do it in a matter of hours. It took years to do. And doing clinical trials for doctors, treating patients. All of that stack is going to change and accelerate.
And speaking of these other fields, actually, for people who are not in science like you are, but maybe like in different fields, what would you advise them about your journey? Like you've been at this for a long time, and thinking about AI and having this curiosity to build with AI every step of the way. What would you like to advise them in terms of how they have to rethink their own field and their own work?
Yeah, so I have one advantage. My job, my work, my life depends on constantly doing experiments. And this is very painful because, especially in biology, 95 to 98% of the experiments you do fail. So I'm very used to failure. That's why it's called experiment. You say, okay, well, try this, try that. And you develop a certain resilience and agency and curiosity just to try. And that's how I was so excited about Codex. And I have to, of course, I showed you the things that worked, but there were a lot of failures before that. Mini apps that I did didn't work very well, and it didn't come out very well. But you shouldn't give up. And my advice to people would be, in the age of AI, really agency and curiosity are the only things that matter. You don't be scared. Constantly try, constantly experiment with AI. Say, what if I can do it this way? But, because you can now ask that what if question. Before, it was just too expensive to ask that. Let's say you have a website for your company. You probably spent thousands of dollars to have it made, and then it wasn't perfect, but you say, okay, well, it's good enough. I don't want to touch it. Now you can say, what if I can just change this and make it this way? Just a matter of minutes, you have a new website or a new product that you can design and then 3D print it, for example. I think everything, this can be applied, but then you have to have that courage to experiment because the cost of experimentation is so low. Why shouldn't you do that?
Yeah.
These are very simple daily things, but I think you can really extend that to pretty much anything in your life. You just have to embrace it and look at AI as something incredibly positive. And I see a lot of negativity. AI is going to do this. AI is going to do that. I believe just the opposite. It's gonna really enhance us to a golden age. And I'm really super excited about it. And I think AI researchers for me are all heroes, by the way, because this is going to be the greatest transformation of humanity. And I'm super excited for the future.
Thank you so much, Derya. Feels like it's amazing, way to close the conversation. Very positive, and we can't wait to see what you're gonna do next and how you're gonna push Codex and these frontier models to bring all of this world together, and work on this digital twin idea.
Absolutely, happy to come back for another episode.
We'd love to have you back in a few months to see how much progress you've made. Until then, enjoy your time in California.
Thank you so much. This was a lot of fun.